So here we are, chapter 10. We are now going to discuss the molecular biology of the gene. So we've spent chapters 8 and 9 going over how how organisms pass on traits. We did that through talking about meiosis and how those traits are expressed. We did that in chapter 9 with genetics. And now we're going to kind of get into how that actually happens. How does the pea plant, right? We know that the homozygous dominant for Mendel's peas is 2 capital P's, which means that those peas carry the purple gene on both of the homologous chromosomes in each one of its cells. But how does the gene for purple turn into actual physical purple pigment? How does that happen? All right, and that's what we want to talk about now. We want to talk about the um, the mechanism by which the cells take the gene and create the purpleness. All right, so brief history. Oh, so let's look at this picture here. We have a deer. We have 3 deer, actually. 2 are brown, 1 is white. Now, if we're talking about this from our genetic standpoint, we would say that deer has albinism. Albinism is usually a homozygous recessive trait. It's homozygous recessive means it inherited those traits from 2 heterozygous parents, more than likely. If we look at this from an evolutionary standpoint, most deer are brown. This deer is white, so the dominant brown color is more prevalent than the recessive white color. Why is that? If you're a coyote, which deer are you going to run after? Or a wolf? You're going to go after the white one because it's easier to see. Right? So we're making these connections. Make the connection between the genetics and the evolution and why it works and why it doesn't work. Right? And that, so this picture says a lot about what we have been talking about. And don't be alarmed if you see this picture again at some point. All right, so 1928. So let's do a brief history of DNA. 1928, Griffiths, a bacteria, oh my word. Griffiths was a bacteriologist, experimented with a bacteria attempting to discover a pneumonia vaccine. And he took back. So Griffiths was attempting to discover a pneumonia vaccine. And what he ended up discovering was when he heat killed the S cells and he mixed them with living R cells, he ended up with dead mice, which means it was some sort of factor that went from the S cells, even though they were heat killed and dead, that the R cells were able to pick up and utilize. The next great discovery came with Avery and McLeod. They declared the DNA was the transforming factor. Now, transforming factor, what does that even mean, right? I always find it fascinating that in 1944 we still didn't know that DNA was the genetic material. So 1944, Avery and McLeod said yes, and the scientific community was like, no. So what they had to do was continue and continue and continue to experiment. 1952, Hershey and Chase is the real moneymaker here. Their experiments with the virus called T2, and T2 infects bacteria. Basically, Hershey and Chase proved that the DNA was the genetic material. So, when the T2 DNA reprogrammed the bacterial host cell, boom, that was it. Right? That's it. They figured out that the radioactive DNA, labeled in green, ended up in the cell and transformed that cell. So, it's huge, right? Once we know that DNA is the transforming factor, we can really start to hone in and focus on DNA because we want to know all there is to know about it. So, who's this young lady, you may ask? This is Rosalind Franklin, and Rosalind Franklin was a woman who did an X-ray crystallography of DNA, combining this picture with These two guys, Watson and Crick, they researched and discovered the structure of DNA in 1953. So 1952, we're like, okay, it's DNA. 1953, we're like, okay, this is the structure of DNA. Now we kind of already know this, right? We know that the nucleotides are made up of three parts: the phosphate, the sugar, and the base. This goes way back to one of our earlier chapters. We know that there are four different nitrogenous bases: A, T, C, and G. So what do we need to know? The phosphate group has a phosphorus atom at the center. The sugar group is a five carbon sugar with 4 carbons in the ring and 1 carbon extending above the ring. The nitrogenous base is a ring of nitrogen and carbon with functional groups attached, and the different functional groups are going to be adenine, thymine, cytosine, and guanine. Now, when we pair up DNA, you'll notice from this picture that A and T pair together, or adenine and thymine pair together, and C and G pair together. So the functional groups A, T, C, G are broken down into different pairings based on their structure. When you look at these, you'll notice that thymine and cytosine, T and C, have one ring, and these are called the pyrimidines. And then A and G have the double ring structures, and they are purines. And when they extend together, there are three rings across. So here are the pyrimidines, here are the purines, right? And when they pair together, ATCG, we end up with 3 rings across. Now, I'm not exactly sure why I included these, so I'm going to just skip over that because we did replication already. So what do we need to know? Right? What do we want to talk about here? We want to talk about protein synthesis. We're trying to get from DNA to purple, right? We're trying to get from the purple protein pigment, from the purple gene to the purple protein, and that's called protein synthesis. So go to your BioFlix and look at the protein synthesis video. It is going to give you a great overview. Watch it before, watch it during, watch it after. So when we're talking about this, right, we're talking about the double helix, we're talking about ATCG, we're talking about mitosis and meiosis, the chromosomes replicating. We know that genotype and phenotype, right? What is that? Now, what's the connection, right? We seem to be getting closer to an answer. The genotype is an organism's genetic makeup. The phenotype is the expression. So we want to get the molecular basis for phenotype is the organism's proteins. So proteins are the phenotype. So genotype is DNA, phenotype is proteins. So we need to get from DNA to protein, and that's going to be through protein synthesis. So on the left-hand side, you'll see a bacterial cell. Transcription and translation are the two processes that we want to make sure that we understand. On the right-hand side is a eukaryotic cell, again with transcription and translation. And those are going to be the two processes that convert DNA information to protein information. This is how we're going to get from the gene that says purple to something that actually is purple. So the steps are as follows. We have transcription, which is the transfer of genetic information from DNA to an RNA molecule. It happens in the nucleus of eukaryotic cells, or in the cytoplasm of the bacterial cell. And it's the synthesis of RNA under the guidance of DNA, right? We have a recipe. The second step is called translation. Translation is the transfer of information of RNA into the protein language. So notice how transcription and translation are both terms that sort of coincide with words. We have the language of DNA. We're going to transcribe that into the language of RNA. Then we're going to translate nucleotide or nucleic acid language into protein language. So we're going to translate. So transcribe and translate. When you transcribe something, you copy it down, either spoken or written word. When something is translated, you take the words of one language and convert them into the same meaning in another language. So that's what's happening with DNA and RNA. The languages of these molecules are converted into the language of proteins. When we talk about DNA and RNA language, the letters are ATCG or AUCG. But when we're talking about proteins, our language is going to be amino acids, right? We got to figure out how we're going to build amino acid language. So we got to figure out how we're going to get through that. And this is it in a nutshell. If you can understand this slide, you can really get there. DNA, obviously a double-stranded DNA. So we're going to use the top as the template strand. We're going to transcribe that into RNA, and we're going to translate that into protein. So the transcription of DNA to RNA, you have to know which strand is going to be the template strand. And basically all you're going to do is pair it up. So when we have a DNA, letter A or adenine, it's going to pair up with a U, a uracil in RNA. When we have a C, it pairs up with G. When we have an A, it pairs up with U. When we have a T, it pairs up with A. And so you just basically make a complementary RNA strand that goes with the DNA strand that you're making a template of. So here, the ACC is going to transcribe into UGG. The second, these are called codons. A codon is a 3 RNA nucleotide sequence. Codon is a code for a specific amino acid, and we'll get there in a minute. So we started with ACC on the blue template strand. We come down a step and we transcribe that into UGG. UGG is a codon that codes for tryptophan, an amino acid. And we'll get to that codon chart in a minute. But hopefully you can see how each of the letters A, C, T, and G will transcribe into U, G, C, and A. I forget what I said, but U, C, G, and A. So we transcribe from DNA to RNA in the nucleus or in the cytoplasm, depending on the cell type. We translate in the cytoplasm both for both different types of cells. All right, so let's get a little bit more in depth with this process. So this is your codon chart. All right, this is there are 64 possible codons because there are 4 different options, A, U, C, and G. So there are 64 possible codons. You'll notice most of them are light purple, but you will also notice a green one as well as 3 orange ones. The green one is the start. It's always going to be the beginning. It's always going to be the first codon of any coded protein. At the end, so that the cell knows where to stop, there were 3 stop codons. UAA, UAG, and UGA are all going to be stop codons, meaning telling the process to stop, translation process to stop. Now, when we talk about codons, remembering that every organism is made up of cells, and every cell at its core has DNA. So A, U, C, and G are the same whether you are a fish, a tulip, a piece of grass, a cat, a human. Doesn't matter. You still have 4 nucleotides to choose from and a variety of different amino acids to choose from. All right, so here we have 64 possible codons. We have all of these amino acids. Remember back from our chapter on proteins, there are only 20 amino acids. So you'll notice here that there are a variety of codons that code for each amino acid. For example, leucine, top left, there are 6 codons that code for leucine. There are 2 for cysteine, there are 6 for arginine, 2 for serine. There's only 1 for tryptophan. So there's no rhyme or reason to it, but there are a number of codons that code for most amino acids. Methionine, there's just 1, and tryptophan, there's just 1. But for the most part, there's a couple. So if we are all A's, T's, C's, and G's, that's where these guys ends up. Now, these are obviously man-made. This is a tobacco plant expressing a firefly gene, making it glow. There's a pig, a yellow-looking pig expressing a jellyfish gene. And basically what that's doing is they've just genetically engineered these animals to have these genes that aren't natural to them, but to show the universality of DNA in that organisms can translate RNA that is not native to them because it's universal. Right? Now, it's a lot more complicated than that. We can't get every cell to transcribe or translate every protein for a variety of reasons. But overall, Every organism is made up of A's, T's, C's, and G's in their DNA, and we have the ability to transcribe and translate almost any of those genes if our cells are programmed to do that. All right, so the property of the genetic code is its universality. So transcription, start from step one. Transcription is when we're going to take DNA and we're going to create a molecule of RNA from it. So there are three phases to transcription. Transcription initiation. When we start the RNA synthesis, there's a nucleotide sequence called the promoter. So here you'll see it as light green. The promoter is the area at the beginning of the gene that has the binding site for the enzyme called RNA polymerase. RNA polymerase in this image is going to be sort of that teardrop looking grayish structure. And that enzyme is going to link RNA nucleotides together. So when the RNA polymerase binds to the promoter, it opens up the DNA, and then it starts making a RNA transcript from the template strand of DNA. It elongates it. The RNA nucleotides come in, they pair up according to ATCG or AUCG. In the elongation step, you'll notice that RNA polymerase is running down the DNA strand. All right, and then the two pieces of DNA come back together. When you're at the very bottom at what's called the terminator, so the termination sequence is when the RNA polymerase reaches a sequence of nucleotides called the terminator. And when it reaches the terminator, that would be the light blue at the end, the RNA polymerase pops off. But what you'll notice is the gene can be used over and over and over again. And now we have this completed RNA transcript. Now, cells can modify the RNA transcript, which we'll look at in a minute. So this is a little bit more of a complete picture. You'll notice that the RNA polymerase still binds to the promoter. It opens up the DNA so it can expose the template strand. In the middle, you see elongation. You'll see the little pink free RNA nucleotides coming in and pairing up. GA, AT, UA, CG, etc. Right, as it sort of reads down, and when we get to the end, the termination, we hit that red terminator DNA. The completed RNA goes, and it's going to be used to be translated, and the DNA can be used over and over again. This is a close-up picture of the RNA polymerase binding to the DNA, opening it up and pairing it up. So the template strand of a given sequence of genes is this. What's the sequence of the non-template strand? So what DNA would match with that, right? It would be ATG, CGG, etc. The mRNA that would match up for this, right, would be AUG, CGG, etc. The resulting amino acid sequence, you would go to the amino acid chart and find those amino acids according to the three nucleotide codon sequence. And then predict how it may fold up. This seems like an odd question. But remember when we were talking about proteins for the first time, we talked about the fact that some of the nucleotides, some of the amino acids were polar, some of the amino acids were non-polar. Polar amino acids are going to attract each other. Non-polar amino acids are going to repel each other. So if you were to draw out the 4 or 5 codon or 4 or 5 amino acids from this, you may be able to see if it's going to bend, fold, attract, repel. So that's pretty interesting. So cells can modify their RNA after transcription. They add what's called a cap and tail sequence. You can see that in yellow. They may splice out certain sections, splicing out of the introns. There might be a poly A tail. There might be, notice how the light pink is taken away and the red is fused together. All right, so this is called modification, RNA modification. And cells do this for a variety of different reasons to come up with the final transcript. When we are ready to translate, we have our transcript, we have processed our RNA and we have our final RNA transcript. Now we're going to go to translation. This is going to happen in the cytoplasm. There are going to be a variety of new molecules here. We have something called a tRNA, which are those green structures. And then we have the ribosome. So mRNA is messenger RNA, that's the red. Transfer RNA, tRNA is the green. Ribosomal or rRNA is the brown. So different types of RNA serve a specific purpose in the translation process. Messenger RNA is what we've just been talking about, the RNA that's coded via DNA and conveys genetic information, right, to translation. So what we need to have is four things. We need to have transfer RNA, we need to have ribosomes. We need to have a chemical energy, here we have GTP, and we need to have enzymes and cofactors. And the only way that these is going to occur is if we have all of those things. Remember that the RNA code is translated into the amino acid sequence. So amino acids don't just line up, right, when they sense the codons. There must be a molecule that tells them what to do. Transfer RNA is what does that. Right? And so you'll see there's a start codon on the bottom left there. That's going to bring methionine. So transfer RNA picks up amino acids and matches them with the appropriate codon. tRNA has two specific sections, right? The anticodon and then the amino acid binding area. And so you'll see here how that tRNA brought the correct protein. And that's what happens during initiation. So initiation is the ribosome comes together surrounding the messenger RNA, the first tRNA, and then from there, we read the mRNA, the tRNAs read the mRNA and bring the various amino acids with them. So initiation is when the mRNA binds to the ribosome, the tRNA binds to the start codon, the initiator tRNA carries methionine to it. The elongation is the incoming tRNA molecule pairs up. The next amino acid is added to the chain through a peptide bond, right? And that's where we get with the dehydration synthesis. tRNA moves over and a spot's open for the tRNA to come in with the next amino acid. And the cycle goes around and around and around and around. And you can see how the chain grows with each addition. Now, when we get to the end, the stop codon, that UAA, UAG, or UGA, when the stop codon is reached, the polypeptides come apart. And now the polypeptide is free to go do what it wants to do. Fold up, be functional, be a purple protein, whatever it wants to do. Everything else, the ribosome, the mRNA, etc., can all be reused. Now this looks complicated, but this is the simplest flow diagram, DNA to RNA to protein. Number one, during transcription, the RNA is synthesized using a DNA template that happens in eukaryotes in the nucleus. Number two, translation begins and each amino acid attaches to a specific tRNA. The first mRNA, tRNA, and ribosome come together. Then the elongation happens where the amino acids are added to the chain. And then there's termination. The polypeptide is ended and released. And from there, the polypeptide can fold up. This is a really, really, really good thing to do for the quizzes, for any question you may have on the final exam, that you should be able to tell me where these various words or terms are used. All right, so take a bunch of these down, figure out where they're used, maybe one place, maybe more than one, maybe only transcription, only translation. All right. And in this question, I always put replication. So this is a great one to quiz yourself on. So does the process of creating protein occur perfectly every time? No. When something goes wrong with a single nucleotide, with the nucleotide sequence of DNA, it's called a mutation. This is sickle cell disease, a single genetic change in the nucleotide sequence from valine, GUA, instead of glutamine, GAA. So this is called a base substitution or a point mutation, where one nucleotide is switched for another. It only affects one amino acid, but it can affect the protein in a variety of ways. In this case, we end up with hemoglobin that does not assume its conformational shape correctly. We can have a silent mutation, a missense, a nonsense, a frame, all those different things that you can have on the left-hand side when you're just doing a substitution, right? Addition, one nucleotide, and it depends on what happens, how it affects the protein. On the right-hand side, we have an addition or deletion. It's called a frame shift mutation. When there's one nucleotide missing, it's called a base deletion, which throws off the entire polypeptide from that point on. And mutations are caused by a variety of things, right? There are spontaneous mutations which occur for no reason. There are physical and chemical agents called mutagens, such as X-rays and UV light. Chemicals, pathogens, HIV, all different kinds of things that can cause different proteins to be created in the cell for a variety of different reasons. So we have mutations, then we have pathogens, but all of these are ways of conveying genetic information to protein. All right, the last thing that we're going to talk about is going to be chapter 11, and this is how we're going to moderate this process. We don't need to create protein all the time in every cell. So how do we fix it? How do we create that? How do we make that start and stop when we need it to?